the size of the vector, which vector is suitable for which type of insert, and the process of transformation in the body of suitable host where it is multiplying, and on the basis of blue and white screening on agar plate in the form of bacterial colonies. Under reproductive cloning, common examples are Dolly, Molly, Polly. And under therapeutic cloning, mainly we dealt with the embryonic stem cells or induced pluripotent stem cells. Cloning vectors in the second session, and expression vectors. Vectors useful for the cloning purpose. How many types of cloning vectors? We already discussed. What are their characteristics? And likewise, expression vectors. On what ground we can say that this is considered as the expression vector? That is only on the basis of three important characteristics: start, ribosomal binding site, and stop codons. Right? Followed by, under the third session, we discussed the screening of colonies, that is blue-white screening. Might be in the white colonies, our gene of interest or desired gene will be present. Now, today we are just starting our sessions with the fourth session, that is purification of protein, protein estimation, followed by the feedback session. So, we are going to start our fourth session, that is purification of protein, followed by the protein estimation. Whatever the clone we have obtained, now we know very well that every gene is expressed in the form of protein. In this case, where clone is already expressed and showing its expression. Now that clone is expressed in the form of a protein. Here, today we have the two sessions, screening of the colonies, that is blue and white screening, because we have to repeat this session again. On that day, n number of participants have not joined. That third session, fourth session, and followed by the fifth session. Third, screening of the colonies, that is on the basis of blue and white screening. Might be our desired gene or gene of interest is in the third session, that is in the white screening, that is our desired gene is present in the white colonies instead of blue colonies. Now, I'm inviting Aditi Parekh, MTech fourth semester student of School of Biotechnology and Autonomous University Teaching Department, Rajiv Gandhi Pradyogiki Vishwavidyalaya, University of Technology of Government of Madhya Pradesh, accredited with grade A by NEC. Aditi Parekh.
Thank you, ma'am, for such a wonderful introduction. And I'll be sharing my presentation within a few moments. So, a very good evening to everyone, and I would like to welcome you to my today's presentation about the blue-white screening. I will explain what it is and how it is performed. So before moving forward, I would like to discuss some of the basics that we have already discussed in great detail in the previous lectures. But for the smooth understanding of the upcoming topic, I would just mention some key points. So first things to start with is this transformation. So what is transformation? DNA transformation is the process during which a plasmid or a vector carrying a specific gene of interest is transformed into another cell, often the bacteria. So we can see here we have a plasmid, that is the circular DNA component found in the bacteria. In all the process, in majority of the cases, the process starts with a procedure to modify the original vector by cloning the target sequence into the polylinker region of the vector. This is done by cutting the vector by restriction enzyme that linearize it in the polylinker region. The target DNA is also cleaved with the same restriction enzyme so that the vector and the gene of interest has the compatible ends. After this, the ends of both these fragments are joined or combined with using the DNA ligase. The goal of this primary step is to modify the original vector by inserting the sequence of insert into the multiple cloning site. This whole process occurs in vitro in a small microfuge tube And if it works properly, we end up with a mixture that contains few copies of the desired vector mixed in with the copies of the cut vector, the DNA fragment, and because the enzymatic reaction is involved in this process, we may or may not get uh, the perfect result. Therefore, there may also be some copies of the original vector present. The next step requires getting the desired vector into the bacterial cell that can then be used to manufacture large quantities of the desired sequence. The strategies used to get the plasmid into the bacterial cell is called transformation. It involves starting with a culture of bacterial cells that have been specifically grown in such a way that it is more likely to take up the plasmid. These cells are called as the competent cells. So how the process is done? The cells are combined with the vector reaction mixture and given a heat shock to create the condition where some of the plasmids are capable of passing up in the cell through the outer membrane. So after the transformation is conducted, the tubes of the competent cell mixture may consist of some of the cells that do not even contain a plasmid because they did not take up any DNA, cells that have taken up the original vector, and others cell that may have the desired vector with the gene of interest inserted into the polylinker site. Moving forward to the next slide. After the transformation, there may be two cases. The bacteria that have taken up the DNA fragment or the original vector and that do not contain the plasmid. So these cells must be selected which contain the plasmid. Identification of these requires the presence of suitable marker genes on the vector molecules whose expression provide a means of identifying cell containing it. Marker genes can be distinguished into two categories. One are the selectable markers or the screenable marker. The selector markers may be the antibiotic resistant sites, as you can see here. This is the ampicillin resistant site. Or the screenable markers are the other kind of markers, like for example the uh, lexi gene present here. A host cell strand is chosen that is selective to a particular antibiotic such as ampicillin or tetracycline or any other suitable antibiotic. The vector used for the recombination purpose are engineered to contain a gene which confers resistance to an antibiotic. After the transformation has been conducted, the cells are plated on an agar medium containing antibiotic to select cells that have been transformed by the vectors. To say whole thing in some simple words, we can say the medium contains the antibiotic agents and the plasmid vector engineered to carry out transformation has antibiotic resistant site. Therefore, the cell which have taken up the vector would survive in the medium, and the cells which do not have vector would die in that medium. Moving to the next slide, the recombinant screening. A selective medium only enables to distinguish between the transformants and the non-transformants. The next problem is to determine which of the transformed colonies comprise cells that contain the recombinant DNA molecule and which contains self-ligated vector molecule. For this purpose, certain screenable marker genes are used, and one technique that incorporates the use of certain screenable marker gene is the blue-white screening, which is a rapid and efficient technique for the identification of recombinant bacteria. Such genes produce enzymes which gives a specific color in the presence of particular substance. For example, in case of the LAGZ gene, it encodes beta-galactosidase, an enzyme that splits lactose into glucose and galactose. Beta-galactosidase also acts on a colorless substance X-gal and develops blue color. The presence of beta-galactosidase, thus to say the presence of the LAGZ gene in the system, can be acid in the presence of X-gal. Now, Now we have to discuss how does blue-white screening work. For screening the clones containing recombinant DNA, a chromogenic substance known as XGL is added to the agar plate. In case if the LEGZ gene is intact, it will produce beta galactosidase, and if beta galactosidase is produced, XGL XGL is hydrolyzed, that is present on the agar plate, is hydrolyzed to form 5-bromo 4-chloroindoxyl, which spontaneously form 5-bromo 4-chloroindoxyl, which spontaneously dimerize to produce an insoluble blue pigment called 5,5-dibromo 4,4 dichloroindigo. The colonies formed by the non-recombinant cells therefore appear blue in color. The colonies will be in blue color and there will be no gene of interest inserted in those bacteria. One thing to pay attention to is the IPTG. IPTG is called as the isopropyl beta D1 thiogalactopyranoside, is used along with the XGL for blue-white screening. IPTG is a non-metabolizable analog of galactose that induces the expression of LEGZ gene, and it should be noted that it is not a substrate for beta galactoside, but it is only an inducer. For screening purpose, we have to add chromogenic substrates like XGL while the screening is being performed. For the screening of the recombinants, with most cloning vectors, insertion of the DNA fragment into the plasmid destroyed the integrity of the one of the marker genes present on the vector molecule. So recombinants can therefore be identified because the characteristic coded by the inactivated marker gene is no longer displayed by the host cell. And this process or this phenomena is called the insertional inactivation. Multiple cloning sites present within the vector molecule in the encodes for the LEGZ gene. This sequence can be nicked by restriction enzyme to insert the foreign DNA. When a plasmid vector containing foreign DNA is taken up by the host, the alpha complementation does not occur. Therefore, functional beta galactoside enzyme is not produced. So no further reaction will take place. And the colonies will observe on the plate will be of white color, and there will be gene of interest inserted within the plasmid of these bacterial cells. So these are the situations we are facing when we are studying both blue colonies and the white colonies on different plate. But this is not the case in the practical conditions. Both blue and white colonies will occur on the same plate. As we can see, these are the blue colonies and these are the white colonies. So the white colonies can therefore be picked up from the plate, and for further purpose they can be cultured in vitro conditions. So this is the pictorial depiction of the downstream processing of how we can extract the protein from the plasmids, or we can store them. But this topic will be discussed in great detail in the upcoming session by Ms. Shivangi, and she'll tell you about each of the steps. I'll just give you the outline, and this will be discussed in a great detail in the upcoming session. We can pick up the white colonies from the plate. We can pellet it down, the bacteria, using column chromatography. Then we can lyse the cell, bind the plasmid to the column, elute the solution out, and store it for the future purpose. So after going through everything, let me summarize the session. So when we get the blue and white colonies on a culture plate, there will be three case scenarios present for the bacterial cells. There will be cells with no plasmid into them. There will be bacterial cells with original vector that have been self-ligated, and there will be cells which contain the vector with the insert. So if the cells are grown in normal LB medium, the first category cell will grow, the second cell will also grow, and the third category of cell will also grow. But if we add ampicillin to the culture media, that is the selective agent, if we add it to the media, then the plasmid or the bacterial cells which do not contain any plasmid, therefore they will not be having any resistance to the antibiotic, so they'll die, they'll not survive. The original vector, the self-ligated vector bacterial cell, will also survive as it will be having resistance because of the antibiotic resistance site, and the vector with the insert will also survive. If we add X gel to the plate, so there will be no bacterial cell containing no plasmid. The original vector will give blue colonies, but they are undesirable to us, so there will be no use of them. And the vector with insert will give white colonies. So these white colonies can be picked up and cultured and thus can be purified, and the protein can be used for further purposes. These are the references. Thank you very much. I hope I can be some help to you. So I would like to invite Ms. Shivangi Srivastav. She is a fellow student in School of Biotechnology to take over the session. Over to you, Shivangi.
Thank you. Thank you very much, Aditi, for inviting me. And your session is very, very good. That is very, very helpful for all of us. Now, I would like to introduce about the next session of this workshop, which is purification of protein. So, before going on the purification step, we have to know what is the basics of protein. So, there is a brief description about the protein. What is that? Proteins are organic macromolecules that are present in all the organisms as well as in viruses. So that is very, very essential organic macromolecules, and it plays an important role in structural and functional fundamentals of life. But sometimes we are thinking just like proteins are mini molecules that having no mass or that having negligible mass. But no, that assumption is wrong. Protein contains 50% dry weight of the cells. So for that, we subdivided, or the scientists have divided into three categories. Like first is globular proteins, which helps in the metabolic reactions. Fibrous proteins, which help to maintain our body structure or elasticity in our muscles, and membrane protein, which is used basically for the cell-cell interaction in cell signaling. And as we all know, proteins are made up of long amino acid chains, which are generally known as peptide chain. So here is the brief description about the peptide chain, that is made up of amino acid, and long polypeptide chain is also known as polypeptide chain. Amino acids are the building block of protein. As like DNA having purines and pyrimidines, as like they are building blocks. As like DNA, protein having building block, that is amino acid. So here are more than 300 amino acids that are known, but only 20 comes in the standard amino acid because it took part in the central dogma process. What is that? Central dogma is first of all DNA replication, then transcription, and then translation. So these the whole process of central dogma having enzymes and proteins. So these proteins and these enzymes that are also a protein takes part in the that processes. So that proteins come in standard amino acid. In the next step, standard amino acid play an important role for growth as well as for our survival for any metabolic reaction in our body. Two amino acids can be linked together by the help of peptide bond, and this reaction is known as condensation process. So here is the description. Like this is the first amino acid, contains the COO group, and this is the second amino acid, which contains in the left NH group, amino group. So condensation takes place when H2O molecule remove out and a form is formed between both the molecule. So there is O is removed from COO minus, and here H2 is removed from NH3 plus. That bond is present. That bond is known as peptide bond, and H2O is released from this. And these both bonds are phi and psi, and their description in between in the Ramachandran plot, which is generally for rotation of any amino acid in 3 dimension. So as I told you about the amino acid, so here is the basic structure of an amino acid, which contains a carbon. That carbon is also known as alpha carbon. From this carbon, here is an H group is present. In CH3 there is a R group, or sometimes H is present in this place. And in the right hand side, COOH, that is carboxylic group, is present. And in the left hand side, NH2 group, that is amino group, is present in the left of the side. When we are going to nomenclate any amino acid, then the counting begins from amino group, not from the C group. So, but in carbohydrate, when we are going to nomenclate any compound, then it will start it from C. But in the case of amino acid, it will start it from the amino group. But as we all know, amino group is not present in this type. It is present in this, which contains both the ion at the same time. That is also known as Rutter ion, and this showing the net ion is 0. When on the pH 7, an amino acid is present in this form. But if you are increasing the pH, it will showing the negatively charged more than positive charge, so it will be alkaline. But if you are decreasing the pH, then it will be showing more and more positively charged. And this is the structure which an amino acid form in 3 dimension. After amino acid, we should know, as I told you, about the side chain, that is alkaline chain, and that because of that alkaline change, an amino acid behavior will be changed. So it will be depend on different type of amino acid, like they will be polar, non-polar, aliphatic, aromatic, negatively charged, and sometimes positively charged. So here are the 20 amino acid which contains all type of bases, like aspartic acid and glutamic acid, which is negatively charged and which comes in polar amino acid, and these are positively charged, which comes in polar amino acid, and these are non-polar amino acid. So what is the difference between polar and non-polar? Polar compounds are those which form a hydrogen bond with the water molecule, and that is also known as hydrophilic. But the non-polar are which are not interested to form a bond between hydrogen molecule, or they don't want any hydrogen bonding of water, with the water. So they are hydrophobic and non-polar. So after this, classification is to be required. So these are basically divided into 2 categories, but sometimes we are talking in 3 categories. That is, first is essential. What are they? Essential amino acid that we need for our growth or for our survival, that our body is unable to synthesize them. So we have to need these amino acid from the external sources. There are 10 amino acid which comes in essential amino acid, but there are second classification which is semi essential amino acid, in which how they correlate with semi or essential. Actually, there are 8 amino acid which comes in essential amino acid, because which are present in both child as well as in adult. But 2 amino acid that are present in adult but not in child, so they comes in semi, because adult body can synthesize them, but a child's body cannot synthesize them. So these are classified into 3. So what is non-essential? Non-essential are those which our body itself synthesize, with the use of or with the help of essential amino acid, or we can say that these amino acids which our body synthesized is the secondary product of essential amino acid. So after knowing the brief description about protein, amino acid and peptide, that when we are going to do any experiment in a lab, we should know why we are doing this. So for our topic, why purification is required of any protein. So purification is required just because just for particular protein. If you want to know the structure and function of a protein, then you should you should be purify that protein for changing its structure and function. As in the last session, sir told you about the vector preparation that is made up of, that is a recombinant DNA technology. In this, then you are going to check a expression of gene, and gene can be expressed in the form of protein, because you can only check the gene expression by its product. So for that product, for that product, we have to check each and everything. So purification is required for that. And obviously, for the mass production of proteins like antibodies, sometimes hormones, enzymes, etc., for their essential use. And to study the protein-protein interaction and protein regulation. As like bait and prey protein, they are interacting each other when they produces a domain on DNA. So for that study, we had to purify our protein, and of course it is for X-ray crystallography, because it produces a structure of tertiary and quaternary, that is in 3D form. So for X-ray crystallography, that is also needed, the purified protein. Otherwise, it will hinder the actual structure of a protein. So before experimenting, you should know the location of a desired product or desired protein. That is, either your protein is present outside the cell, or inside the cell, or in the transfuse in the membrane. So why we need this knowledge of location? Because if your protein is present outside the cell membrane, then there is no need of cell disruption. There is no need of cell disruption technique, and it will you will take with the help of enzymatic or chemical treatment easily, without disrupting any cell and without disturbing any cell. But if your protein present in the transfuse form, only membrane would be degraded. No any organelle membrane, no any nuclear membrane will be degraded. And if your protein present inside the cell, then various techniques would be used for purification. Why? Because you had to purify your product from different impurities. So here's some steps from starting to the end. When you produces a vector, then then you would prepare the media as just that is suitable for that vector or that organism. After that, fermentation takes place, cell harvesting, and these three steps come in the upstreaming processes. After that, cell debris removal, that comes in downstreaming processes, protein solution concentration, final purification, and the last one is product. So our topic is purification of protein. So we should concentrate only on downstreaming processes, not in upstreaming processes, because there are so many research and development and for that. So we have to discuss only about the protein purification. So first step is protein isolation steps. There are 3 steps from which we can isolate a protein. First is solubilization of protein. Solubilization can be done by the cell disruption processes. There are so many processes which are used in cell disruption. The second one is stabilization of proteins. If your protein is not stable, then it will be no use for our experiment. And the last one is purification is required. So the first step is solubilization of protein. We are using many techniques for cell disruption, from which The first one is ultrasonication. We can use French press also for solubilization of protein. We can use enzymatic from isolating a protein from the outside or inside of the cell. And chemical methods can be used. And freeze thawing, homogenizer, bead mills, thermal, and the last one is centrifugation. These methods you can use for the solubilization of protein. After solubilization, stabilization of protein is required. Why it is required? Because it is required because you should maintain the structure of that protein. So for maintaining the structure, pH should be monitored and temperature should be maintained, because these two factors can affect the normal protein's structure. So the last, for maintaining the structure, we have to precipitate our protein by the help of salting out method. What is that? It is a dialysis method. It's like dialysis bag in which salt and protein both are present, and that membrane is semi-permeable. Only small particles can move out and in, and this works on the principle of osmosis. So for that, precipitation is required, because if there is no precipitation, then your protein can be degraded. And so here is the definition of sorting out. Sorting out is a purification method that utilizes the reduced solubility of certain molecules in a solution of very high ionic strength. And sorting out is typically, but not limited to, the precipitation of large biomolecules like proteins. So after stabilization, the last step is purification of proteins. So purification can be done by the help of chromatography, and it is used to separate a desired product from the mixture. And it is used for the separation of amino acids, proteins, and carbohydrates. It is used for the analysis of drugs, hormones, and vitamins, and very helpful for the qualitative and quantitative analysis of complex number. And technique is used also. The very most important use of chromatography is the determination of molecular weight of any protein. So what is the principle of chromatography? Chromatography consists of two phases. First one is mobile phase and second one is stationary phase. So here is the concept of adsorption which takes place in the principle. So what is mobile phase? Mobile phase refers to the mixture of substance to be separated or dissolved in a liquid or gas. Your mobile phase can be liquid or can be gas. So for that, your mixture that you want to isolate or that you want to purify, which will be dissolved in the mobile phase. What is a stationary phase? That is a porous solid matrix through which the sample contained in the mobile phase percolates. That process is known as adsorption. The interaction between the mobile phase and the stationary phase that results in the separation of a compound from a mixture or separation of an amino acid from the mixture. So here are some types of chromatography in graphy for using for our purification technique, that is paper chromatography, thin layer chromatography, gel, column, ion exchange, gel filtration, and affinity. So before discussing about the chromatography, I would like to say that paper chromatography and thin layer chromatography that are not used for the protein purification technique, but it contains the normal basic of chromatography from which you can correlate your next chromatography. So what is paper chromatography? Wait, I'll discuss with the diagram. In paper chromatography, you can take a paper, that is chromatographic paper, or you can say that it is a filter paper. Okay, so here is the solvent, and here is the this jar is sealed. After before sealing this jar, you add your mixture from which you want to isolate, or mm, you purify, that you want to purify that product. So that is basically used for the pigment collection. After, when this solvent moves in the in the from the paper, then it will rises and it will show these differences in the colors. And RF is the retention factor from which we can calculate which product is this. When why, how? Distance traveled by the component, this, and distance traveled by the liquid. If you want to calculate about this, what is that? Then if you calculate this, then you can check from the standard table of retention factor, from which you can correlate which product is this, either it is chlorophyll A, chlorophyll B. So this is the basic concept of chromatography, how it is separated. After that, thin layer chromatography contains the adsorption principle. So what is that? It is just similar to paper chromatography, and it contains a stationary phase. In that, we use only paper, so there is no stationary or mobile phase, only there is a paper. But in this case, we use a slide, glass slide, in which we added adsorbent, that is thin in the thin layer, and that adsorbent are the like silica gel, alumina, and cellulose, and that becomes the stationary phase. So when you put your sample here, it will move in the forward direction with the solvent. Then it will separate it in the different part. And this is a lab photo. So I put this in. When you can see the difference in the color. So compared to why it is used, it has the advantage of faster run, better separation, and the choice between different adsorbents. So here's the first column chromatography, which is used conventionally. So what is that? It is a separation technique which contains a stationary bed within a tube. And if you simply wants to understand it, we use a column which is looking like a burette, but its thickness or its diameter is more than a burette. So that column is used for the that column is used in which we added we produce a bed stationary bed, which is made up of adsorbents like silica, alumina, charcoal powder. Sometimes we are using soda lime for that, because after that, this stationary phase leaving an open unrestricted path of the mobile phase in the middle of the tube. Wait, I'll discuss with the diagram, then you can understand it very easily. Here is the diagram. As I told you, it is looking like a burette and it containing a bulb. In the first, this is the bed, stationary bed, and here you can load your sample or mixture. Then when it moves during the gravity or due to the gravity, then it will separate out with the in the different bands. Like you can see that the strong interaction, and these are the weaker interaction. So when it comes here, you can collect it separately and identify which product is this that you collected. So ion exchange chromatography is the newly method which is used for the protein interaction, in which what they use is like simple. I don't want to go for theory. Like it is here, you can see this is a column. This column contains positive or negative charge. Why? Because it contains this process. Here you can use exchange resins, which is cation exchanger or anion exchanger. So if you want to purify your amino acid which containing any charge, like you want to isolate the positively charged, then how can you negatively charge or positively charge? Then here will be the opposite charge. As you all know, opposite charge attract each other and same charge repel each other. So for that concept, they use anion exchanger or cation exchanger as a resin in the stationary phase, from which here they flow the mixture of amino acid, from which positively in this diagram you can see easily, then they both opposite charges producing the ionic bond between them, and the left one which having the same charges repelling and come out from the column. Then you can isolate these purified protein from this method. Gel filtration chromatography having so many names that you can say is a size exclusion chromatography, and also known as gel permeation chromatography, gel filtration, and it is also referred as molecular sieving or molecular exclusion chromatography. But it is used it can separate molecules according to their size, their shape, and their molecular weight. Smaller molecules are able to enter the pores of the media, and therefore molecules are trapped and removed from the flow of the mobile phase. However, molecules that are larger than the average pore size of the packing are excluded and thus suffered essentially no retention. Such species are the first to be eluted, or that we can say that are waste. This is how the molecules are separated. Here is the diagram of this chromatography. In this, they use gel as like for electrophoresis. When we place our DNA in the gel, then it will move according how it will move from the pores. So as like same concept we are using for gel filtration. These are the gel, and these molecules that are bigger than that pore size, so they will elute first and they are waste. But if the size of these molecules is just equal or is less than these pores, then it will travel from this, and after they eluted. So these eluted particles are the purified particle, and these are the waste particles. So you can use this chromatography uh, which can purify a protein, or there is no need of polishing or the last step for purification. It is also used here is the useful here is the uses of that. That is, it is used for the determining the tertiary structure and quaternary structure of purified protein, especially since it can be carried out under native solution conditions. After gel filtration, there is affinity chromatography, which having the principle is same as like ion exchange. In ion exchange, we are using ions on the stationary phase, but in affinity, we used sometimes ligands when we are purifying the receptors. We can adhere or we can attach in the stationary phase that substrate for enzyme purification, and sometimes we are attaching the antigen for antibody purification. So what is that? Affinity chromatography is based on selective non-covalent interaction between an analytic and specific molecules referred to as ligands. Here is the example of ligand, but you can correlate this example with enzymes as well as for antibody. The immobilized ligands act as molecular